So I want to start off just by asking you what the biggest shift for you was, or the biggest learning for you was in completing your PhD. And it may be completely unrelated to the topic you did your PhD on, maybe a lesson around, the philosophy of science, or a personal lesson, or it may be related.
So my PhD focused on basically tying together early research showing that chronic infection, so infection with bacterial pathogens, viral pathogens, fungal pathogens, and chronic infection with many of those organisms probably contributes to a number of diseases that we call autoimmune. And also that the activity of those single pathogens, for example, a single viral or bacterial pathogen, also have a profound impact on the activity or balance of the sort of collective organism ecosystems in our bodies, the human microbiome. And so one of the greatest takeaways I had is that when you are looking at inflammation in a condition or in a person, it is very important to start to tie that inflammation to the activity of the organisms in the human body, or to some of the infectious agents and pathogens that we unfortunately become infected with.
How come that's important? Curious if you could elaborate a little bit there.
It's very important for treatment is the main reason, because if you think for example, in ulcerative colitis and Crohn's disease would be a good example. These are GI diseases that are characterized by inflammation of the intestine or the gut, right? And early on, research teams were just measuring that inflammation. And with that model where you're just measuring the immune response and saying, yes, there's a lot of inflammation going on here, what you do for treatment is that you basically shut down or stifle the immune response. So what became the standard of care for patients with those illnesses were immunosuppressive drugs that knock down T cell function or the activity of other immune cells in those patients. But a little after that, teams began to collect samples from those same patients and say, wait, we're learning that there are more and more and more organisms interacting communities of organisms in the human gut, basically the human microbiome and virome, which are, trillions of interacting bacteria, viruses, and fungi that have these rich communities in the gut and the intestine, and now many other parts of the human body, we know. And so teams began to say, are the activity of those organisms connected to the inflammation we're measuring in Crohn's disease and ulcerative colitis? And now that research is moving forward. And what happens is that the therapeutics change, because now you can target potentially the organisms that may be actually at the root of that inflammation instead of having to completely shut down the immune response itself. And that leaves the person in much better shape, because those immunosuppressive drugs have really bad side effects, because otherwise you really don't want a depleted immune system, right? So in other words, by going to that sort of root cause level that may be organisms at the root of inflammation in chronic disease, you can come up with treatments that are less immunosuppressive and less harmful for the patient.
That makes total sense. So it allows you with precision to address the root cause rather than just to sort of try and shut down the whole system. And as a result of doing that, potentially help the symptoms a little bit, but also, knock down everything else that's good.
Exactly, because that's one of the things, for example, if you're an ulcerative colitis patient and you get on the immunosuppressive drugs, you're basically on them for life. You need to take them and manage them. And often people require higher doses to keep that inflammation down. But there's now potential that if you address the microbiome that may be at the root of inflammation, you may actually be able to just improve that, get off the medication, get back to a life where you don't always need to be on medicine. So yeah.